The surface of the ocean is a world of light, motion, and familiar boundaries. But as we leave the sunlit waves behind, the water deepens, colours fade, and the temperature plummets. We're descending into the abyss, a realm of perpetual darkness where the rules of terrestrial biology no longer apply. Here, the weight of the entire ocean presses down with immense force, a crushing environment that should theoretically restrict life to the smallest, most minimalist forms. Yet in this freezing void, nature presents a profound paradox. Instead of shrinking, many creatures expand to monstrous proportions. Today we will explore why deep-sea creatures become giants. In these extreme depths, single-celled organisms swell to the size of tennis balls, and invertebrates that are tiny on land grow longer than human beings. This biological mystery challenges everything we understand about survival, adaptation, and energy conservation. Why does a habitat with almost no food and absolute darkness produce the largest invertebrates on Earth? Is this terrifying scale a mistake of evolution, or is it the ultimate strategy for survival in the deep? If you enjoyed diving into the abyss with us, feel free to leave a like and subscribe for more deep-sea science. And before you go, drop a comment letting us know where in the world and what time it is as you are watching this video. Welcome to the frontier of oceanography, where the laws of nature are turned upside down and the true rulers of the planet remain hidden beneath miles of water. To truly comprehend the mystery of deep-sea gigantism, we must first establish a rigorous definition of this biological phenomenon. Deep-sea gigantism, scientifically known as abyssal gigantism, is an evolutionary trend where certain species of marine invertebrates, fish, and other organisms grow to significantly larger sizes than their close relatives inhabiting shallower coastal waters. This is not a random occurrence, nor is it isolated to a single evolutionary branch. Instead, it is a widespread convergent evolutionary strategy observed across completely different phyla, including arthropods, mollusks, echinoderms, and chordates. To understand the scale of this phenomenon, we must look at the physical environments that separate the surface world from the deep abyssal plains. In shallow marine ecosystems, solar radiation drives primary production through photosynthesis. Coral reefs, kelp forests, and mangrove swamps are packed with energy, allowing for rapid life cycles, intense competition, and high biodiversity. However, because energy is highly contested and predators are numerous, smaller organisms often rely on agility, camouflage, or high reproductive rates, rather than sheer body mass. When we descend past the bathypelagic zone into the abyss, which lies between 4,000 and 6,000 meters below the surface, the environmental parameters shift dramatically. Sunlight is completely absent. The temperature hovers just above freezing, typically between 1 and 4 degrees Celsius. Food resources are incredibly scarce, consisting primarily of detritus falling from the upper layers of the ocean. In a habitat defined by such extreme deprivation, classic biological models suggest that animals should minimize their body mass to reduce energy consumption. Smaller bodies require fewer calories to maintain basic cellular functions. Yet, the deep ocean repeatedly breaks this expectation. Species that are diminutive, almost microscopic in shallow waters, transform into true titans when isolated in the deep sea. To appreciate the sheer biological divergence caused by abyssal gigantism, we can perform a direct anatomical and physiological comparison between shallow water organisms and their deep-sea counterparts. Consider the order Isopoda, a diverse group of crustaceans. In shallow terrestrial and aquatic environments, we encounter common isopods known as woodlice, pillbugs, or sea slaters. These creatures are typically small, measuring anywhere from a few millimeters to 2 or 3 centimeters in length. They have delicate segmented exoskeletons, simple compound eyes, and minimal internal space for resource storage. Their life expectancy is short, their metabolic rate is relatively high for their size, and they must constantly forage for decaying vegetation or organic matter to sustain their daily energy requirements. Their anatomy is optimized for a fast-paced life cycle in an environment where food is readily available, but predators are abundant. Now, let us look at the deep-sea counterexample, the giant isopod, scientifically classified under the genus Bathynomus. Living at depths exceeding several hundred to thousands of meters, these abyssal titans can reach lengths of over 40 to 50 centimeters and weigh more than 1.5 kilograms. Anatomically, the giant isopod is not just a scaled-up version of a pill bug, it is an entirely re-engineered organism. Its exoskeleton is heavily calcified, forming a thick armoured vault that protects its internal organs from physical damage and ambient pressure. Its internal anatomy features a massive, highly distensible digestive tract, capable of storing immense quantities of food for long periods. Unlike its shallow water relatives that must eat constantly, the giant isopod can survive for several years without consuming a single meal, owing to its vast internal storage capacity and an incredibly reduced basal metabolic rate. We see an identical pattern of anatomical amplification within the class Amphipoda. In coastal rock pools and sandy beaches, amphipods, commonly called sandhoppers or scuds, are nearly invisible to the naked eye. They seldom exceed 1 or 2 centimeters in length. They flit rapidly through the water column or burrow into sediment, consuming microscopic algae and organic debris. Their bodies are fragile, and their physiological systems are designed for rapid nutrient turnover. If we travel down to the Mariana Trench, the deepest point on the planet, we find the giant amphipod, Alicella gigantea. This extraordinary creature can reach lengths of 34 centimeters, making it more than 20 times larger than its shallow water relatives. The anatomical differences are profound. The deep-sea giant amphipod possesses highly specialized enzymes capable of breaking down tough, complex organic compounds that shallow water species cannot digest. Furthermore, its muscular and circulatory systems are structurally reinforced at a molecular level to maintain functional integrity where ordinary tissue would fail. This divergence is even more pronounced when examining the phylum Mollusca, particularly the cephalopods. In coastal waters, squids are generally modest in size, with most species, such as the market squid or the European squid, measuring between 20 and 30 centimeters. They are lightning-fast, high-energy predators that rely on rapid, oxygen-intensive jet propulsion to catch prey and evade predators. Their metabolic rates are exceptionally high, demanding constant nourishment, and their lifespans are brief, often lasting only 1 to 2 years. Their eyes are highly developed for detecting motion in sunlit waters, and their muscular mantles are thin and highly flexible. When we descend into the deep sea, we encounter the true apex invertebrates of the planet. The giant squid, Architeuthis ducks, and the colossal squid, Mesonychoteuthis hamiltoni. These biological marvels can reach lengths of 12 to 14 meters and weigh up to 500 kilograms. The anatomical differences between a common coastal squid and a colossal squid represent a complete evolutionary paradigm shift. The muscular mantle of the deep-sea giant is much thicker and contains higher concentrations of ammonium chloride in its tissues, which provides neutral buoyancy, allowing the creature to float effortlessly in the water column without expending precious kinetic energy. Its circulatory system features a massive multi-chambered heart configuration designed to pump blood under low-temperature conditions. Its nervous system is dominated by the largest eyes in the animal kingdom, measuring up to 30 centimeters in diameter. This anatomical adaptation allows the giant squid to capture the faintest traces of light, including bioluminescence from prey or the silhouettes of predatory sperm whales, a sensory capability completely unnecessary and absent in shallow water species. What driving biological mechanisms cause these stark structural variations? Anatomists point to several key factors, including cell size, growth periods, and indeterminate growth patterns. In many shallow water species, biological maturation is achieved rapidly due to warmer temperatures and high predation pressure. Once reproductive maturity is reached, energy is redirected from somatic growth toward gamete production, causing the organism to stop growing. In contrast, deep-sea organisms experience delayed sexual maturity. Because the environment is stable, cold, and largely devoid of seasonal fluctuations, these creatures can afford to spend decades growing before they ever reproduce. This extended juvenile and subadult phase allows their skeletal structures and muscular tissues to continuously expand over time. Additionally, the anatomical composition of deep-sea giants often reflects a phenomenon known as gigantocellularity, where individual cells within the organism's body grow larger rather than merely multiplying in number. Larger cell volumes can be highly advantageous in cold, stable environments because they reduce the overall surface area of cell membranes relative to internal cellular volume. This structural shift minimizes the energy required by the organism to run ATP-driven ion pumps, which maintain chemical gradients across cell membranes. In a shallow water organism, a vast amount of daily energy is consumed simply keeping these cellular pumps running at high speeds. The deep-sea giant, by adopting larger cells and a vastly increased overall body volume, eliminates this metabolic tax, allowing it to utilize its structural mass as a giant battery for long-term survival. Ultimately, comparing shallow and deep-sea organisms reveals that deep-sea gigantism is not a deformity or an ecological fluke. It is a highly sophisticated, multi-layered biological adaptation. Where the shallow water organism is built for speed, rapid reproduction, and high energy turnover within a volatile ecosystem, the deep-sea giant is anatomically engineered for ultimate efficiency, structural endurance, and resource conservation within a timeless frozen void. Every anatomical change, from the thickening of a crustacean's carapace to the enormous expansion of a cephalopod's eye, serves to turn the harsh limitations of the abyss into a definitive evolutionary advantage. To truly appreciate the existence of deep-sea giants, we must explore the intense physical forces acting upon their bodies. The most defining, unrelenting, and potentially destructive environmental factor of the deep ocean is hydrostatic pressure. For every 10 meters we descend beneath the surface of the sea, the pressure increases by approximately 1 atmosphere, which is equivalent to the weight of the air pressing down on us at sea level. At the average depth of the abyssal plains, which lies around 4,000 meters down, the pressure reaches 400 atmospheres. At the ultimate depths of the ocean trenches, such as the Challenger Deep within the Mariana Trench, the pressure escalates to over 1,000 atmospheres. This is a force exceeding 100 megapascals, equivalent to having an adult elephant standing on the tip of a human thumb, or trying to withstand the crushing weight of 50 commercial jumbo jets resting upon a person's chest. For a long time, early marine biologists believed that life simply could not exist under such conditions. They assumed that the immense physical force would crush any complex biological structure into oblivion. However, this perspective was built on a fundamental misunderstanding of physics and fluid dynamics. Water is highly incompressible. Because the bodies of deep-sea organisms are composed primarily of water, they do not collapse like an empty aluminum soda can underfoot. The internal pressure within their tissues matches the external hydrostatic pressure of the surrounding ocean perfectly. There are no air-filled cavities such as lungs or traditional swim bladders, which would otherwise implode under the strain. By eliminating compressible gas pockets entirely from their anatomy, deep-sea giants achieve a state of physical equilibrium with the abyss. Yet, even without air spaces, hydrostatic pressure poses a much more insidious microscopic threat to life. At the molecular level, extreme pressure exerts a powerful, destabilizing effect on the fundamental building blocks of organisms: lipids, proteins, and cellular membranes. Under hundreds of atmospheres of pressure, water molecules are forced closer together, altering their physical behavior. This increased molecular density interferes with the chemical bonds that hold complex biological molecules together. Consider the structure of cellular membranes. In shallow water organisms, cell membranes are composed of a fluid bilayer of phospholipids. This fluidity is absolutely vital for life. It allows nutrients to pass into the cell, waste products to exit, and membrane-bound proteins to move and interact freely. However, when subjected to the extreme pressure of the deep sea, these phospholipid bilayers undergo a phase transition. The pressure forces the individual lipid molecules into an incredibly tight packed configuration, effectively freezing the membrane. It transforms a fluid dynamic barrier into a rigid crystalline solid. When a cell membrane solidifies, the transport proteins embedded within it become immobilized. These cells can no longer pump essential ions, transmit electrical signals, or absorb nourishment. For a shallow water animal, this sudden cellular immobilization leads to immediate physiological failure and death. To combat this crushing effect, deep-sea giants have evolved a profound biochemical adaptation known as homeoviscous adaptation. Instead of using the standard saturated fatty acids found in shallow water organisms, deep-sea creatures construct their cell membranes using incredibly high concentrations of polyunsaturated fatty acids. These unsaturated fats possess bent, kinked molecular tails that prevent them from packing tightly together under pressure. By incorporating these flexible molecular structures into their membranes, deep-sea giants ensure that their cellular barriers remain highly fluid, pliable, and fully functional, even under the weight of an entire ocean. An even greater challenge occurs within the structure of proteins and enzymes. Life is entirely dependent on the specific, three-dimensional shapes of proteins. Enzymes must flex, bend, and change shape to catalyze chemical reactions, replicate DNA, and convert nutrients into metabolic energy. The shape of a protein is held together by delicate, weak chemical bonds, such as hydrogen bonds and hydrophobic interactions. Under intense hydrostatic pressure, water molecules are violently forced into the internal cavities of these protein structures. This hydration process disrupts the internal bonding, causing the complex three-dimensional proteins to unfold, lose their shape, and break apart. This catastrophic structural failure is known as protein denaturation. Without functional enzymes, all biochemical pathways grind to a halt. How do deep-sea giants prevent their essential proteins from unraveling under this extreme stress? The answer lies in a fascinating group of protective molecules known as piezolites. Piezolites are small, organic osmolyte molecules that act as chemical stabilizers within the cellular fluid. The most prominent and well-studied piezolite in deep-sea organisms is trimethylamine N oxide. Trimethylamine N oxide works by altering the structural behavior of the water molecules surrounding cellular proteins. It acts as a powerful water structuring agent, effectively forcing water molecules to form a tight, organized network around themselves, rather than invading the delicate interior of the proteins. By excluding water from the internal cavities of enzymes, trimethylamine oxide creates a protective chemical shield that reinforces the protein's structural stability. This allows the enzymes of deep-sea giants to retain their precise three-dimensional configurations and continue catalyzing life-sustaining reactions under hundreds of atmospheres of pressure. Interestingly, scientists have discovered a direct linear correlation between the depth at which an organism lives and the concentration of trimethylamine N oxide within its muscle tissues. The deeper a creature descends, the more of this protective compound it accumulates. This biochemical reality explains why deep-sea fish caught from great depths often possess a distinct, pungent, fishy odor. The smell is a direct result of the breakdown of the very molecules that kept their cells from collapsing. This molecular stabilization provides a direct pathway toward understanding deep-sea gigantism. Maintaining stable proteins and fluid cell membranes under extreme pressure requires a significant expenditure of evolutionary energy. Every single cell must continuously produce stabilizing molecules and synthesize complex unsaturated lipids just to stay intact. If an organism is small, it has a high surface area to volume ratio, meaning it has a large amount of exposed cellular surface relative to its internal mass. This structural reality increases the relative energetic cost of defending against hydrostatic pressure at the cellular boundary. By growing into a giant, an organism dramatically decreases its surface area to volume ratio. A larger body volume means that a smaller proportion of the animal's total cellular mass is directly interacting with the boundary forces of the environment. This geometric reality allows deep-sea giants to optimize their internal biochemical efficiency. Furthermore, the stabilization of structural proteins by compounds like trimethylamine N-oxide allows for the construction of larger, more robust connective tissues and muscular networks that do not degrade over time. The structural matrix of the animal's body becomes incredibly stable. Thus, the crushing hydrostatic pressure of the abyss acts as a powerful evolutionary sieve. It systematically eliminates any species that lacks the precise biochemical tools required to protect its molecular architecture. For those species that do evolve homeoviscous membranes and high concentrations of piezolites, the deep ocean ceases to be a crushing prison. Instead, it becomes a realm of unparalleled structural stability, providing a vast, unyielding physical canvas upon which nature can sketch the grand outlines of its largest, most resilient masterpieces. Beyond the immense physical force of hydrostatic pressure, deep-sea organisms must confront another severe environmental challenge: the profound, unchanging cold of the abyss. Below the thermocline, which is the transitional layer where ocean temperatures drop rapidly, the deep sea enters a state of thermal stagnation. Across the global abyssal plains, temperatures remain constantly low, typically hovering between 1 and 4 degrees Celsius. This freezing environment is uniform, stretching across millions of square kilometers of the ocean floor. In such a cold habitat, the laws of thermodynamics dictate the survival strategies of every living creature, acting as a direct evolutionary driver toward large body sizes. To understand how deep-sea gigantism serves as an adaptation against extreme cold, we must look at Bergmann's rule. Formulated originally to describe terrestrial warm-blooded animals, this ecological principle states that within a broadly distributed taxonomic group, populations and species of larger size are found in colder environments, while populations and species of smaller size are found in warmer regions. While the physiological mechanisms differ between warm-blooded endotherms and cold-blooded ectotherms, the underlying geometric and thermodynamic principles remain identical. It is a matter of surface area to volume ratios. Every living organism is a biochemical system that generates and exchanges heat and energy with its surrounding environment. The rate at which an organism loses heat or interacts thermally with the surrounding water is directly proportional to its surface area, which is the total amount of skin, shell, or tissue exposed to the external world. Conversely, the capacity of an organism to retain energy, store nutrients, and maintain internal metabolic stability is directly proportional to its volume, which represents the total mass of its internal tissues and cellular structures. As an object or an organism increases in size while maintaining the same shape, its volume grows much faster than its surface area. This mathematical relationship means that a large animal has a much smaller surface area relative to its total body mass than a small animal. In the freezing waters of the deep sea, where the ambient temperature is just above the freezing point of water, a small organism with a high surface area to volume ratio loses its precious internal thermal energy rapidly to the surrounding environment. Its small body acts like a highly efficient radiator, dissipating energy into the infinite thermal sink of the ocean. For a giant organism, however, the geometric advantage is substantial. Because its massive volume is shielded by a relatively small external surface area, the rate of passive energy loss is greatly reduced. The core of the animal's body becomes thermally insulated by its own structural mass. Even in cold-blooded invertebrates, which do not maintain a constant elevated body temperature like mammals do, this thermal inertia is critical. A larger, bulkier body slows down the rate of chemical and thermal equilibrium with the freezing water. This stabilization allows internal metabolic reactions to proceed with greater consistency, shielding delicate cellular machinery from sudden localized thermal fluctuations. Furthermore, the freezing temperature of the deep sea has a profound effect on the kinetics of biochemical reactions. According to the Van 't Hoff rule and the concept of metabolic temperature coefficients, the rate of chemical reactions, including those that drive animal metabolism, decreases by half for every 10 degrees Celsius drop in temperature. In the 1 degree Celsius water of the abyss, the cellular machinery of an organism naturally slows down to a fraction of the speed seen in warm surface waters. Muscle contraction becomes sluggish, nervous system conduction delays increase, and the synthesis of essential molecules slows down. To maintain functional mobility and perform essential survival tasks like hunting, scavenging, or escaping danger in slow-motion conditions, an organism requires a robust physical structure. A small, delicate creature operating at such low kinetic speeds would be highly vulnerable, unable to generate enough mechanical force to overcome the viscosity of cold water. Water becomes denser and more viscous as its temperature approaches freezing. For a tiny organism, moving through cold water feels like swimming through thick syrup. Deep-sea giants solve this physical problem through sheer muscular and structural scaling. By developing massive bodies, large limbs, and elongated muscles, they generate substantial mechanical leverage. The absolute force produced by a larger muscle mass easily overcomes the increased viscosity of the freezing water, allowing giant isopods, massive sea spiders, and large deep-sea fish to glide purposefully across the ocean floor. Their large scale transforms an environment that would immobilize a smaller creature into a stable, manageable arena. Additionally, the combination of large body size and cold temperatures alters the growth dynamics of these animals. In warm surface waters, high temperatures accelerate the life cycle. Organisms grow rapidly, mature early, reproduce quickly, and die young. In the freezing deep, the low kinetic energy of the environment delays the onset of sexual maturity. Deep-sea organisms grow at an exceptionally slow pace, but they continue growing for a very long time. Because they do not expend their energetic resources on early reproduction, they can channel their limited nutrient intake entirely into somatic growth over decades. This prolonged, uninterrupted growth period allows them to bypass the size limitations that restrict their shallow water relatives. Ultimately, the 1 to 4 degrees Celsius temperature of the deep ocean acts as a patient architect of giants. By favouring organisms that maximize their internal volume while minimizing their exposed surface area, the cold environment rewards structural scale. Deep-sea gigantism is a masterclass in thermodynamic optimization. It proves that in the vast, freezing dark of the abyss, being large is not a display of evolutionary excess, but an elegant, geometrically sound strategy for conserving energy, overcoming physical resistance, and achieving long-term biological stability. To truly understand how deep-sea giants conquer the immense physical boundaries of the abyss, we must examine their underlying metabolic architecture. In the deep ocean, energy is the ultimate currency, and it is in extraordinarily short supply. Because no sunlight penetrates past the upper 200 meters of the water column, photosynthesis is impossible throughout the vast majority of the ocean's volume. The deep-sea floor is an ecological desert, dependent entirely on a sparse, unpredictable rain of organic debris, falling from the sunlit surface thousands of meters above. To survive in this environment of chronic starvation, deep-sea giants have abandoned the high-energy lifestyles of their shallow water relatives. Instead, they have mastered a profound evolutionary strategy, the art of living in ultra-slow motion. At the heart of this survival strategy is an extreme reduction in the organism's basal metabolic rate, which is the baseline amount of energy required to keep a body alive while at rest. Deep-sea giants possess some of the lowest metabolic rates ever recorded in the animal kingdom. This physiological slowdown is beautifully described by Kleiber's law. This biological principle states that an animal's metabolic rate scales to the 3/4 power of its body mass. This means that as an organism grows larger, its energy consumption per unit of body mass drops significantly. A large animal is exponentially more fuel efficient than a small one. By scaling up their body mass to gigantic proportions, deep-sea creatures exploit this law to its absolute limit. A giant isopod or a colossal squid requires a fraction of the weight-specific calories that a shallow-water crustacean or coastal squid demands. Their bodies function like highly efficient, ultra-low power machines. Every internal process, from cellular repair to blood circulation, is dialed down to a minimal operational baseline. Their hearts beat only a few times per minute. Their nervous systems transmit signals at a measured, deliberate pace, and their physical movements across the seafloor are slow and calculated. This minimal energy expenditure ensures that the few calories they do consume are not wasted on unnecessary activity. This ultra-low metabolic rate allows deep-sea giants to transform their massive bodies into highly efficient energy storage banks. In a habitat where weeks, months, or even years can pass between significant meals, the ability to store vast reserves of nutrients is the difference between survival and extinction. When a food source does appear, such as a large fish carcass or a fallen marine mammal, deep-sea giants engage in opportunistic, highly efficient feeding. Because their internal anatomy is scaled up, they possess massive, highly distensible digestive tracts. They can gorge themselves, consuming several times their own body weight in a single feeding event. Once a meal is consumed, the low metabolic rate ensures that these nutrients are processed with extreme care and stored long-term. Deep-sea giants do not immediately burn through their food to fuel active lifestyles. Instead, they convert the excess energy into massive deposits of high-density lipids and oils, which are packed throughout their muscular tissues, livers, and specialized storage organs. These lipid reserves act as an internal biological battery. Because their baseline daily energy requirement is so close to zero, these massive fat stores can sustain a deep-sea giant for astonishing periods of time. Laboratory observations of giant isopods have revealed that these creatures can survive for over 5 continuous years without a single meal, showing no visible signs of tissue degradation or systemic failure. A smaller organism, with its fast-paced metabolism and minimal storage capacity, would burn through its energy reserves and starve to death within days under the same conditions. Beyond the scarcity of food, deep-sea giants must also navigate the severe physiological challenge of oxygen limitation. In many regions of the global ocean, beneath the productive surface zones, lies a massive layer of water known as the oxygen minimum zone, which typically extends from depths of 200 down to 1,000 meters. Within this zone, the decomposition of falling organic matter by bacteria consumes vast quantities of dissolved oxygen, depleting the water until it contains only a tiny fraction of the oxygen found at the surface. Even below this zone, in the true abyssal depths, water circulation can be incredibly slow, leaving the environment hypoxic, meaning it is chronically low in dissolved oxygen. For a high-energy surface animal, entering such an oxygen-deprived environment causes immediate suffocation. Oxygen is the essential electron acceptor that drives aerobic respiration, the cellular process that produces the vast majority of an organism's energy. Without it, cellular metabolism collapses. Deep-sea giants, however, have evolved highly sophisticated physiological modifications that allow them to extract every available molecule of oxygen from the surrounding water, ensuring their cells never experience starvation. First, deep-sea giants possess exceptionally large, highly efficient respiratory surfaces. The gills of giant deep-sea crustaceans and the respiratory structures of deep-sea mollusks are vastly expanded in surface area compared to those of shallow water species. These gills are structurally designed to maximize contact with the surrounding water, ensuring that even the most minute traces of dissolved oxygen are captured. Furthermore, the blood chemistry of these animals is completely overhauled. Deep-sea giants utilize highly specialized respiratory pigments, such as hemocyanin in crustaceans and octopuses, or hemoglobin in deep-sea fish, that have evolved an extraordinarily high affinity for oxygen. These specialized proteins bind to oxygen molecules with immense strength at the respiratory surface, refusing to release them until they reach the deep internal tissues that require them most. Once inside the body, The slow-motion metabolism ensures that the captured oxygen is used with maximum efficiency. Because their cells are not working at high speeds, their oxygen demand is continuously low. If oxygen levels drop even further, many deep-sea giants can seamlessly transition into anaerobic metabolic pathways, producing energy through chemical processes that do not require oxygen at all. They can tolerate high levels of metabolic waste products such as lactic acid that would be toxic to surface creatures. By combining high-affinity respiratory chemistry with a low-demand cellular lifestyle, these giants navigate oxygen-depleted waters with absolute ease. The combination of an ultra-low metabolic rate, massive energy storage capacities, and highly efficient oxygen management yields an extraordinary biological byproduct, a vastly extended lifespan. In the history of biology, there is a clear inverse relationship between metabolic rate and longevity. Organisms that live fast and burn through energy rapidly tend to age quickly and die young. Their high-speed metabolic processes generate vast quantities of cellular waste products, particularly reactive oxygen species, which are highly destructive molecules that damage DNA, degrade proteins, and disrupt cellular membranes over time. This continuous molecular damage causes rapid aging and short lifespans. Deep-sea giants, by choosing the path of absolute metabolic minimalism, generate only a fraction of these destructive cellular byproducts. Their internal molecular environment is incredibly stable and clean. Because their cells operate at such a low kinetic speed, the wear and tear on their genetic and structural machinery is reduced to a minimum. Furthermore, their bodies produce high levels of advanced cellular repair enzymes that patiently fix what little damage does occur over time. This lack of internal decay, combined with the extreme stability of the cold deep-sea environment, allows these creatures to achieve record-breaking lifespans that dwarf those of surface animals. Consider the Greenland shark Somniosus microcephalus, a massive deep-sea predator that inhabits the cold deep waters of the North Atlantic. Growing up to more than 6 meters in length, this shark exhibits an exceptionally slow growth rate, expanding by only about 1 centimeter per year. Its metabolic rate is among the lowest of any vertebrate, and its movements are slow and deliberate. Using advanced radiocarbon dating techniques on the crystalline proteins within the lenses of their eyes, marine biologists discovered that the Greenland shark can live for at least 400 years, making it the longest-lived vertebrate on the planet. They do not even reach sexual maturity until they are roughly 150 years old. We see a similar trend among deep-sea invertebrates. Giant tube worms living near cold seeps and certain deep-sea clams, such as the quahog clam, have been confirmed to live for hundreds of years. These animals do not experience the rapid systemic decline associated with aging in surface species. Their lives are measured not in years, but in centuries. Ultimately, the low-energy, slow-motion lifestyle of deep-sea giants is a masterclass in evolutionary compromise. In the abyss, trying to maintain a high-speed energetic existence is a direct path to starvation. By shrinking their metabolic demands to a bare minimum, expanding their internal nutrient storage, adapting their blood chemistry to low oxygen environments, and extending their lifespans over centuries, these magnificent creatures have turned a habitat of extreme deprivation into a realm of long-term security. They prove that in the grand design of nature, speed and aggression are not the only paths to dominance. In the silent dark of the deep ocean, the slow, the efficient, and the massive are the true masters of survival. To fully appreciate the mechanics of deep-sea gigantism, we must look beyond generalized physics and conduct a deep anatomical audit of specific species that exemplify these rules. Our first case study is the legendary giant Isopod, an animal that looks like a prehistoric relic but is actually a masterpiece of modern deep-sea engineering. Thriving at depths of over 2,000 meters, where the light is completely dead and the physical force of the water column approaches 20 megapascals, these massive crustaceans have completely overhauled their outer anatomy to handle the stress of the abyss. The most striking feature of the giant isopod is its armor. In shallow water relatives, the exoskeleton is thin, flexible and vulnerable. In the giant isopod, the exoskeleton has become a heavily calcified vault. This armour is made of a complex composite matrix of chitin, which is a tough structural carbohydrate, reinforced with high concentrations of calcium carbonate and calcium phosphate. This biochemical mineralization process hardens the shell until it mimics the density of stone, creating a structural shield that is highly resistant to bending, cracking, or collapsing under hydrostatic pressure. This armoured carapace is organised into 14 overlapping segments known as tergites, which cover the thorax and abdomen. This segmented layout is essential. If the giant isopod possessed a single, solid, unyielding shell like a turtle, the crushing pressure of the deep sea would concentrate forces on specific structural weak points, causing the shell to shatter. By distributing its mass across 14 distinct interlocking plates, the giant isopod creates a flexible shield. When external pressure presses down on the animal, the overlapping plates slide slightly against each other. This movement distributes the physical force evenly across the entire surface area of the body, preventing dangerous stress points from forming. Beneath this hard shell, the giant isopod's legs, or periopods, are engineered for heavy-duty stabilization. It has 7 pairs of legs, each tipped with sharp, highly calcified claws. These limbs are short, stocky, and positioned low to the ground. This low-profile stance reduces the animal's centre of gravity, which helps it remain stable when walking over loose abyssal mud. The internal joints of these legs are lined with thick pads of specialised pressure-resistant cartilage that prevent the bones of the armour from grinding together under the weight of the water column. This combination of calcified armour, segmented stress distribution, and stable legs allows the giant isopod to crawl over the deep ocean floor without expending precious energy fighting the physical forces of its home. While the giant isopod represents the ultimate mastery of structural defence on the ocean floor, the giant squid, Gigantism ducks, represents a radical transformation in open water sensory evolution. Moving through the midnight zone between 1,000 and 3,000 metres deep, this apex invertebrate has evolved a sensory feature that stands completely alone in the history of biology: an enormous visual system. The eye of a giant squid can reach diameters of up to 30 centimetres, making it roughly the size of a standard basketball, with a lens as large as an orange. No other animal on Earth, including the blue whale, possesses an eye of this scale. To understand why the giant squid evolved such massive eyes, we must examine how light behaves in the deep ocean. Past a depth of 200 metres, sunlight fades fast. By the time we reach 1,000 metres, the ocean is dark and downwelling sunlight is completely gone. However, the deep ocean is not entirely black. It is illuminated by bioluminescence, which is chemical light generated by living organisms. Over 75% of deep-sea creatures, including fish, jellyfish, and small invertebrates, can produce their own light to communicate, attract mates, or confuse predators. Ordinary eyes, like those of shallow water squids, are built to detect high-contrast images in bright sunlight. In the deep sea, those small eyes are useless because they cannot capture enough photons to create a clear image. The giant squid's eye is engineered as an extreme low-light collector. The internal layout of the eye features an incredibly wide aperture and a massive pupil that can dilate to maximum width. This large opening allows the eye to gather the absolute maximum amount of light from the surrounding water, collecting even the faintest flashes of bioluminescence. Behind the pupil sits a large, highly spherical lens that focuses this captured light onto a massive, high-density retina. The retina of the giant squid is packed with millions of specialized, elongated photoreceptor cells called rhabdomeras, which are highly sensitive to the blue and green wavelengths of light that travel farthest through seawater. This density of light-sensitive cells gives the giant squid a level of visual sensitivity that is unmatched. It does not look for high-definition shapes. Instead, it detects faint, moving gradients of light against the black backdrop of the abyss. The primary evolutionary purpose of this massive visual system is survival, specifically the detection of its ultimate predator, the sperm whale. Sperm whales are massive, warm-blooded mammals that dive down into the deep ocean to hunt giant squids. As a sperm whale swims through the dark water, its large body inevitably collides with thousands of microscopic bioluminescent organisms, such as dinoflagellates and small jellyfish. This physical disturbance causes these organisms to flash, creating a glowing wake of blue light that follows the whale through the dark. A small eye would never see this faint glow from a distance, but the giant squid's basketball-sized eyes can detect this moving wake of bioluminescence from over 100 meters away. This advanced visual warning gives the squid a few extra seconds to prepare its defense. It can immediately cease all movement, flatten its body to reduce its profile, or use its muscular siphon to fire a jet of water, escaping into the darkness before the whale can strike. Furthermore, the giant squid has integrated this visual system with a complex network of internal balancing organs known as statocysts. Located inside the cartilaginous skull near the brain, statocysts are fluid-filled chambers containing small stones made of calcium carbonate, called statoliths. As the squid moves, changes orientation, or detects the physical vibrations of an approaching predator, these stones roll over sensory hairs inside the chamber, sending immediate updates to the brain. By combining these vibration-detecting statocysts with its massive eyes, the giant squid creates a dual sensory grid that tracks both light and motion in a world of total darkness. When we look at these two animals side by side, we see how deep-sea gigantism takes completely different forms depending on the lifestyle of the organism. For the giant isopod, a slow-moving scavenger on the seafloor, gigantism is expressed as a heavily armoured, calcified vault built to withstand high hydrostatic pressure and protect its internal storage systems. For the giant squid, a free-swimming predator in the open water column, gigantism is focused into an oversized visual and sensory system designed to read the faint light signals of the deep sea and avoid predators. Both animals prove that in the abyss, growing larger is not a random evolutionary accident. It is precise, targeted strategy that changes the vulnerabilities of a hostile environment into a permanent survival advantage. To understand the incredible diversity of deep-sea gigantism, we must examine our next two case studies: the Japanese spider crab, Macrochera kempferi, and the oarfish, Regalicus glesni. These two creatures represent completely opposite structural approaches to growing large in the deep ocean. While the Japanese spider crab relies on a massive, heavy articulated exoskeleton to navigate the sea floor, the oarfish utilizes an incredibly elongated, paper-thin and ribbon-like skeletal design to float effortlessly through the water column. Both species demonstrate that the physical constraints of the deep sea can be solved through completely different engineering pathways. We begin with the Japanese spider crab, an ancient marine arthropod that holds the record for the largest leg span of any living animal on Earth. Found on the deep continental shelf and upper slopes of the Pacific Ocean, particularly around the waters of Japan at depths ranging from 300 to 600 meters, this giant crustacean can achieve a staggering leg span of up to 3 and sails 0.8 meters from the tip of one claw to the other. Its central body, or carapace, can grow to 40 centimeters in width, and the entire organism can weigh up to 19 kilograms. The anatomy of the Japanese spider crab is a marvel of biomechanical engineering. Its legs are incredibly elongated, looking like the spindly limbs of a terrestrial spider, but they are composed of thick, heavily mineralized tubes of chitin reinforced with calcium compounds. This length provides a critical locomotion advantage on the soft, muddy sediments of the deep-sea floor. Instead of wading through the deep mud, which would require an immense amount of energy and cause the animal to sink, the Japanese spider crab uses its long, stilt-like legs to walk gracefully over the terrain. Its weight is distributed over a massive surface area, preventing it from sinking into the substrate and allowing it to move efficiently while foraging for food. However, possessing a massive exoskeleton comes with a catastrophic biological tax, the requirement of molting, or ecdysis. All arthropods must periodically shed their hard outer shells in order to grow larger, but for a giant operating under the immense pressure and cold temperatures of the deep sea, this process is an incredibly dangerous event. The metabolic and physical mechanics of a Japanese spider crab molt represent one of the most stressful events in the animal kingdom. The molting process begins weeks before the actual shell is shed. Under the influence of specific hormonal signals, the crab begins to absorb calcium and other minerals from its old exoskeleton, storing them within its internal tissues. This process weakens the old shell, making it brittle. At the same time, the underlying epidermal cells begin secreting a new, soft, wrinkled cuticle beneath the old armour. Because the deep sea temperature is so low, this biochemical synthesis of a new lining proceeds at a slow pace, demanding a significant portion of the animal's baseline energy reserves. When the moment of ecstasis arrives, the giant crab must find a stable, flat area on the seafloor. It absorbs vast quantities of seawater into its circulatory system, inflating its internal volume. This hydrostatic inflation creates immense internal pressure that forces the old carapace to split open along a preformed structural line at the back of the body. The crab must then slowly, carefully, back out of its old shell. It must pull all 10 of its long limbs out of their narrow, rigid tubes without snapping or damaging them. In the cold water of the deep ocean, this physical extraction can take several hours of continuous muscular exertion. If a single leg becomes trapped within the old shell, the crab will fail to free itself and will die of exhaustion or be eaten by predators. Once the crab successfully extracts itself from its old armour, it enters its most vulnerable state. It is a soft-bodied giant. Its new exoskeleton is completely pliable, resembling the texture of soft leather. To achieve its new, larger size, the crab must immediately pump more seawater into its tissues, stretching the new shell out to its maximum dimensions before it hardens. The mineralization process, where calcium is deposited back into the new chitin matrix to harden it, takes days to complete in the cold abyss. During this critical window, the giant crab cannot stand or defend itself. It must remain perfectly still, hidden in the dark, relying on its low metabolic rate to conserve oxygen, while its new armour cures. This high-risk process proves that deep-sea gigantism requires a massive investment of physiological energy and structural resilience. If the Japanese spider crab represents the ultimate expression of heavy armoured skeletal design, the oarfish represents the absolute peak of lightweight, elongated minimalism. Inhabiting the mesopelagic zone down to depths of 1,000 metres, the giant oarfish is officially recognised as the longest bony fish alive on the planet. It can reach confirmed lengths of over 8 metres, with unconfirmed reports stretching up to 11 metres, yet its body width is incredibly thin, often measuring only a few centimetres across. It looks like a massive silver ribbon gliding through the dark water. The skeletal structure of the oarfish is a radical departure from standard fish anatomy. To survive in the deep ocean column without expending precious energy on constant swimming, an organism must solve the problem of negative buoyancy. Most shallow water fish use a gas-filled swim bladder to maintain their position in the water, but at great depths, maintaining a gas pocket requires complex chemical engineering and runs the risk of implosion under pressure. The oarfish solves this problem by completely eliminating the swim bladder and overhauling its entire skeleton. The bones of an oarfish are not heavy and dense like those of shallow water fish. Instead, they are incredibly light, porous and fragile, containing a high concentration of water and low levels of heavy minerals. The skull and vertebral column are reduced to a minimalist framework of flexible cartilage and thin bone. Furthermore, the entire body of the oarfish is highly compressed laterally, meaning it is paper-thin from side to side. This extreme flat shape reduces its physical mass to a fraction of what a round fish of the same length would weigh. By shedding this structural weight, the oarfish achieves neutral buoyancy naturally, allowing it to float in the water column without sinking. This ribbon-like anatomy dictates a unique method of locomotion known as amorphic swimming. Instead of flexing its entire body side to side like a shark or a salmon, which would require a massive expenditure of muscular energy, the oarfish holds its long body perfectly straight and vertical in the water column. It propels itself by undulating a long, bright red dorsal fin that runs the entire length of its back. This fin contains hundreds of individual delicate rays that move in a continuous rhythmic wave. This movement allows the oarfish to hover in place, moving slowly up and down through the water column with minimal muscular effort. The oarfish often hovers in a vertical head-up position, pointing its face toward the surface of the ocean. This posture is connected to its specialized feeding strategy. Despite its terrifying sea serpent-like appearance, the oarfish is a gentle filter feeder. It does not possess large teeth or a powerful jaw. Instead, its mouth is small, highly protrusible, and lined with delicate structures known as gill rakers. By hovering vertically in the dark, the oarfish looks upward, using its large eyes to detect the silhouettes of small krill, tiny crustaceans, and jellyfish drifting above it. When a swarm of prey is detected, the oarfish opens its mouth, expanding its oral cavity to create a powerful vacuum that sucks the prey inside, where they are trapped by the fine mesh of its gill rakers. Even the skin of the oarfish is adapted for this lightweight, low-energy existence. It completely lacks traditional protective scales, which would add unnecessary weight to its body. Instead, its silver skin is covered in a specialized compound called guanine, which forms a reflective metallic surface that bounces back the faint traces of bioluminescent light in the deep ocean, acting as a dynamic form of camouflage. If a predator does manage to attack, the oarfish possesses a unique ability known as autotomy. It can safely drop or break off the trailing end of its long-tail section to escape danger, sacrificing a portion of its non-essential body mass while keeping its vital internal organs, which are packed into the front section of its body, safe. When we analyze these two species together, we see the incredible flexibility of evolutionary adaptation. The Japanese spider crab and the oarfish have taken completely different paths to achieve giant status. The crab chose a heavy, armoured stilt-leg design to conquer the flat mud of the seafloor, accepting the high danger of the molting process as the price of its scale. The oarfish chose an ultra-lightweight, paper-thin, ribbon-like structure to drift through the open water column, trading physical strength for perfect energetic efficiency. Both animals confirm that in the deep ocean, there is no single formula for success. Whether through armoured mass or minimalist length, deep-sea gigantism is an elegant calibration of anatomy against the forces of the abyss. To fully comprehend the horizontal expansion of deep-sea gigantism, we must leave the barren abyssal mud and venture into two of the most extreme, highly specialized environments of the ocean, the boiling hydrothermal vents and the endless open water column of the midnight zone. Here, we encounter two organisms that redefine the very nature of biological growth, tissue engineering, and individuality. These are the giant tube worm, Riftia pachyptila, and the massive colonial Siphonophores. While the giant tube worm achieves immense size by forging a microscopic chemical alliance with bacteria, the Siphonophore grows into a Leviathan by linking thousands of individual cloned organisms into a single, cooperative superorganism. We begin with the giant tube worm, a biological anomaly that thrives along the volcanic cracks of the ocean floor, primarily along the East Pacific Rise, at depths of over 2,500 meters. In these hydrothermal vent zones, tectonic plates tear apart, allowing magma to superheat the surrounding seawater to temperatures exceeding 350 degrees Celsius. This superheated fluid is packed with toxic heavy metals and high concentrations of hydrogen sulfide, a chemical compound that is highly lethal to almost all surface life because it permanently halts cellular respiration. Yet, directly inside this toxic plume, giant tube worms grow at a speed that matches or exceeds almost any other marine invertebrate on Earth. reaching lengths of over 2.5 meters within less than 2 years. The anatomy of the giant tube worm is a study in radical evolutionary reduction. It has no mouth, no eyes, no stomach, no gut, and no anus. It possesses a completely closed, blind structural design. It cannot chew, ingest, or physically digest food in any traditional sense. Instead, the entire secret to its massive scale lies within two highly specialized anatomical structures: the bright red respiratory plume at the top of its body, and a large internal organ known as the trophosome, which fills the interior of its white chitinous tube. The bright red plume, which extends into the mix of cold seawater and hot vent fluid, is packed with an extraordinary network of vascular blood vessels. The deep red colour is caused by unique, highly complex hemoglobin molecules that are vastly larger and more structurally resilient than human hemoglobin. While human blood can only transport oxygen, the specialised hemoglobin of the giant tube worm possesses distinct, separate binding sites that allow it to carry oxygen, carbon dioxide, and highly toxic hydrogen sulfide simultaneously through its circulatory system without poisoning the animal's own tissues. The blood pumps these chemicals down into the trophosome organ. The trophosome is essentially a massive biological factory, accounting for up to 60% of the worm's total internal body weight. This tissue is packed with billions of symbiotic sulfur-oxidizing bacteria. This relationship represents a complete departure from the solar-driven energy cycle of the surface world. Instead of relying on photosynthesis, these specialized bacteria perform a process known as chemosynthesis. Inside the trophosome, the bacteria absorb the hydrogen sulfide and oxygen delivered by the worm's blood. They chemically break down the hydrogen sulfide, stripping electrons from the molecule to generate chemical energy in the form of adenosine triphosphate. The bacteria then use this generated energy to fix inorganic carbon dioxide into complex organic molecules such as sugars, amino acids, and lipids. This metabolic production is highly efficient. The bacteria produce far more organic nutrients than they require for their own survival, leaking the excess directly into the bloodstream of the giant tube worm. In return, the tube worm provides the bacteria with a safe, stable home and a continuous high-volume supply of essential raw chemicals. By building its body around this internal chemical factory, the giant tube worm bypasses the scarcity of the deep sea, unlocking a massive, continuous stream of energy that allows its protective white cylinder to expand to giant proportions. While the giant tube worm achieves gigantism through internal chemical collaboration, the Siphonophores achieve monumental scale by manipulating the boundaries of cellular individuality. Moving through the pelagic zone between 1,000 and 4,000 meters deep, certain species of deep-sea Siphonophores, such as Praia dubia or Apolemia uvaria, can reach lengths of 40 to 50 meters. This makes them significantly longer than the blue whale, placing them among the longest continuous animal structures on the planet. However, calling a Siphonophore a single animal is scientifically inaccurate. A Siphonophore is a highly complex, free-floating colonial organism composed of thousands of distinct individual bodies called zooids. Every single zooid within the colony is genetically identical, created through a continuous process of asexual budding from a single larval egg. But instead of separating to live independent lives, these zooids remain physically linked, sharing a common continuous digestive and nervous pathway that runs like a central highway down the entire length of the colony. To achieve functional efficiency at such a massive scale, the Siphonophore employs an extreme form of physiological division of labour. Each individual zooid is highly specialised, modifying its anatomy to perform only one specific function for the survival of the greater whole. It is a biological assembly line operating in the open water column. At the very top of the massive chain sits a single gas-filled float, known as the pneumatophore, which acts as a buoyancy regulator. Directly beneath the float is a cluster of specialized propulsion units called nectophores or swimming bells. These zooids are shaped like translucent bowls and contain powerful muscular walls. They do nothing but pump water, contracting rhythmically in perfect unison to propel the massive winding chain through the deep water column. They possess no mouth, no reproductive organs, and no sensory systems. They are pure living engines. Extending behind this propulsion cluster is the Siphosome, a long stem that can stretch for tens of meters through the dark. Arranged along this stem are repeating units of specialized zooids focused on nutrition, defense, and reproduction. The feeding units, known as gastrozoids, are equipped with a single mouth and a highly distensible stomach. When food is captured, these gastrozoids digest the prey and pump the liquefied nutrients into the central shared circulatory canal, distributing energy to the swimming bells and other non-feeding parts of the colony miles away. For defense and prey capture, the colony relies on dactylozoids. These units extend long, hair-like tentacles that can hang down into the water column like a massive drift net. These tentacles are packed with millions of highly specialized, explosive, stinging cells called nematocysts, which inject powerful, paralyzing neurotoxins into any fish or small crustacean that brushes against them. Many deep-sea siphonophores modify these defensive units to display bright, rhythmic, bioluminescent flashing, using the chemical light as a lure to draw curious prey directly into the invisible wall of stinging cells. Finally, the colony contains gonozoids, which are units dedicated solely to producing eggs and sperm to ensure the generation of new colonies. This configuration allows the siphonophore to overcome the primary constraint that limits the size of ordinary solitary animals in the open ocean: the problem of structural scaling and mass. A single solid animal measuring 50 meters long would require an immense heavy skeleton and a massive muscular network to coordinate its movements, demanding a high-energy metabolic output that the deep sea cannot support. The siphonophore solves this through its modular, decentralized design. Its body is composed of a lightweight gel matrix that matches the density of seawater perfectly, meaning it weighs almost nothing in terms of dry mass. It does not fight the water, it drifts through it like a living net. If a section of the chain is broken or severed by a predator, the colony does not bleed to death or experience systemic failure. The remaining segments simply continue operating, immediately budding new zooids to replace the lost pieces. When we look at the giant tube worm and the colonial siphonophore side by side, we see how deep-sea gigantism completely breaks the traditional rules of biology. The giant tube worm proves that an animal can shed its entire digestive tract and grow to monumental proportions simply by transforming its inner tissues into a thriving volcanic city for chemosynthetic bacteria. The siphonophore proves that an organism can shatter the traditional concept of an individual body, linking thousands of specialized clones into an ultra-long cooperative net that dominates the open water. Both creatures confirm that in the vast, resource-starved spaces of the deep ocean, the most successful paths to giant scale require a radical departure from individual isolation, replacing it with the absolute perfection of biological integration. To fully comprehend the horizontal development of deep-sea gigantism, we must examine the trophic dynamics that fuel these massive bodies. In an ecosystem where primary production via photosynthesis is completely absent, the delivery of energy relies entirely on gravity. The structural scale of deep-sea giants is directly tied to two distinct gravitational phenomena that act as nutrient delivery vehicles from the sunlit surface to the benthic dark. These are the slow, continuous descent of organic matter known as marine snow, and the sudden, catastrophic influx of massive biomass known as a whale fall. Together, these two resource streams dictate the structure of the abyssal food chain, providing the necessary calories to sustain the largest invertebrates on Earth. We begin with marine snow, which serves as the fundamental baseline food supply for the global deep ocean floor. When viewed through the cameras of an oceanographic submersible descending through the midnight zone, the water column appears to be filled with an endless, drifting blizzard of white particles. This is not frozen water. It is a complex organic matrix composed of dead phytoplankton, decaying zooplankton hulls, empty shells made of silica and calcium carbonate, fecal pellets from surface fish, and mucus feeding webs discarded by soft-bodied pelagic organisms. As these microscopic pieces sink at a slow rate of only a few tens to hundreds of meters per day, they are continuously broken down and recycled by midwater bacteria. By the time this organic material escapes the upper ocean and lands on the abyssal plains 3,000 to 6,000 meters below, over 99% of its original surface energy has been stripped away. What remains is a highly depleted, nutrient-poor sediment that covers the seafloor like a vast mud desert. To survive on this sparse, low-quality diet, small organisms must spend an enormous amount of metabolic energy filtering massive volumes of water or mud just to extract a few calories. For a tiny creature, this constant physical exertion is a losing battle that severely restricts growth. Deep-sea giants, however, have evolved highly specialized anatomical modifications that allow them to exploit this continuous rain of marine snow with ultimate efficiency, transforming a poor resource into a stable fuel source for long-term growth. Many abyssal giants have become expert detritivores, meaning they consume this decaying organic matter directly from the sediment. Giant sea cucumbers, which can grow to over 0.5 meter in length, move slowly across the muddy floor like massive biological vacuums. Their mouthparts are ringed with large, highly branched feeding tentacles covered in a sticky mucus coating. They sweep these tentacles over the surface layer of the mud, precisely capturing the newly fallen marine snow before it is mixed into the deeper, sterile sediment. Their highly extended digestive tracts are designed to process massive volumes of low-nutrient mud slowly, extracting the bound organic compounds with powerful, highly specialized digestive enzymes. Because their movement across the ocean floor is measured in centimeters per hour, and their basal metabolic rate is incredibly low, the energy extracted from this slow diet easily exceeds their daily operational cost. The excess calories are systematically stored within their thick gelatinous body walls, allowing them to maintain their large physical size year-round in a habitat defined by chronic nutrient scarcity. While marine snow represents a reliable but low-energy background diet, the second resource stream, a whale fall, represents a sudden, explosive concentration of pure energy that completely rewrites the local ecology of the deep sea. When a massive marine mammal, such as a blue whale or a sperm whale weighing 40 to over 100 metric tons, dies near the surface of the ocean, its immense body eventually loses buoyancy and begins a rapid, heavy descent to the seafloor. As it strikes the abyssal plain, it introduces a massive island of pure lipid, protein and bone tissue into an ecosystem that has not seen such a concentration of nutrients for centuries. A single whale carcass can deliver an equivalent volume of organic energy to a specific patch of the seafloor that would normally take up to 2,000 years to accumulate via the slow drift of marine snow. The sudden arrival of this massive biological prize triggers an immediate, highly organized ecological succession that can last for more than a century, split into 4 distinct, scientifically recognized phases. Each phase is dominated by a completely different assemblage of organisms, with deep-sea giants playing a critical role in the initial exploitation of the resource. The first phase is known as the mobile scavenger phase, and it begins within minutes of the whale carcass landing on the ocean floor. The physical impact of the carcass sends a massive chemical plume of scent molecules drifting through the horizontal current layers of the deep ocean. Deep-sea giants, equipped with highly developed chemosensory receptors, detect this signal from kilometers away. From all directions across the dark desert, giant isopods, massive amphipods, and large abyssal hagfish begin to crawl and swim rapidly toward the epicenter of the feast. During this initial phase, which can last from several months to up to 2 years depending on the size of the whale, the mobile scavengers strip the soft tissue from the bones. Giant isopods use their strong, highly calcified mouthparts to tear away large chunks of skin and muscle tissue. Their highly expandable stomachs allow them to consume several times their own body weight in a single feeding event, swelling into rigid, ball-like shapes. Once filled to capacity, they crawl away into the nearby sediment, entering a state of complete metabolic dormancy, where they slowly process the concentrated lipids over many months. This opportunistic feeding strategy allows them to secure enough high-density fuel from a single whale full to power their growth and reproduction for several years, bypassing the limitations of the surrounding desert. Once the soft flesh has been entirely stripped away, the ecosystem transitions into the opportunistic phase, which lasts for an additional 2 to 5 years. During this period, the sediment surrounding the bones becomes heavily enriched with organic fats and oils that have leaked out of the decomposing carcass. The site is colonized by millions of smaller, highly specialized worms and crustaceans. While these opportunistic species are smaller in scale, they form a dense, high-volume living carpet that draws in larger invertebrate predators, creating a localized, highly productive food web that operates at a rapid pace. The third phase is the most scientifically astounding and is known as the sulfophilic or chemotrophic phase. This stage can persist for several decades, driven entirely by the decomposition of the whale's massive skeletal system. Whale bones are composed of up to 60% high-density lipids by weight. As anaerobic bacteria slowly break down these deep fats inside the porous matrix of the bone, they consume the remaining oxygen and release massive quantities of dissolved hydrogen sulfide into the surrounding boundary water layer. This chemical transformation effectively turns the old skeleton into a temporary hydrothermal vent system. The surface of the bones is colonized by thick mats of chemosynthetic bacteria that oxidize the toxic hydrogen sulfide to produce organic energy. These bacterial mats provide a rich food source for giant deep-sea limpets, snails, and specialized dune worms, creating an autotrophic ecosystem that is completely independent of the surface world. This long-term concentration of chemical energy allows deep-sea organisms to maintain a highly stable community for generations, utilizing the slow decay of the bone matrix as a permanent habitat. Finally, after all the organic fats have been extracted from the bone matrix, the site enters the reef phase. What remains of the skeleton is a sterile, heavily calcified structure made primarily of calcium phosphate. In a habitat defined by soft shifting mud, these hard bones provide a critical structural anchor for sessile filter-feeding organisms. Deep-sea corals, large glass sponges, and massive anemones attach themselves to the stable surface of the bone, utilizing the elevated structure to extend their feeding apparatus into the passing horizontal currents, capturing the faint drift of marine snow. When we look at the interaction between marine snow and whale falls, we see the true brilliance of the deep-sea food web. The evolutionary survival of deep-sea giants is not built on a steady daily intake of food. Instead, it is built on a highly sophisticated economic model of resource management. Marine snow provides the predictable, low-volume baseline energy required to maintain their ultra-low metabolic systems, while they rest in the cold dark. The sudden, chaotic arrival of a whale fall provides the massive concentrated lipid capital required to fuel their large physical structures, expand their carapaces, and complete their reproductive cycles. By engineering their bodies to survive on absolute minimalism during the long periods of drought, and developing the sensory tools to rapidly exploit the moments of absolute abundance, these magnificent titans prove that in the grand design of the ocean, the scarcity of an environment can be entirely overcome through the absolute perfection of structural efficiency and opportunistic patience. To fully explore the horizontal expansion of deep-sea gigantism, we must leave the physical parameters of the modern laboratory and step back into the cultural history of human ocean exploration. Long before oceanographic submersibles and remote-operated vehicles began capturing high-definition digital footage of the abyssal plains, human beings were already interacting with the magnificent titans of the deep. These encounters did not take place in controlled scientific environments. Instead, they occurred at the volatile boundary of the ocean surface, where dying, injured, or disoriented deep-sea giants occasionally breached the sunlit world. To ancient mariners who possessed no knowledge of hydrostatic pressure, homeoviscous adaptation, or metabolic scaling, these sudden, terrifying apparitions could not be explained by natural science. Instead, they were absorbed into human mythology, serving as the biological blueprints for the most famous sea monsters in history, the Kraken and the Leviathan. The transition from biological reality to maritime myth is most clearly seen in the legend of the Kraken, a monstrous cephalopod said to haunt the freezing waters of the North Sea, particularly around the coasts of Norway, Greenland and Iceland. For centuries, Scandinavian fishermen recorded terrifying encounters with a creature of immense scale, describing it as an island-sized beast with dozens of long, snake-like arms lined with sharp hooks. According to historical naval logs, the creature would emerge from the dark depths to encircle wooden merchant vessels, using its muscular limbs to crush the hulls and drag entire crews down into the watery abyss. We now know with scientific certainty that the biological catalyst for the Kraken myth was none other than the giant squid, Architeuthis ducks, and its polar relative, the colossal squid. While these animals spend their entire adult lives swimming in the complete darkness of the mesopelagic and bathypelagic zones, specific oceanographic disruptions can force them to the surface. When a deep-sea squid experiences a parasitic infection, systemic neurological failure, or is violently caught in a powerful upwelling thermal current, it can lose its ability to maintain neutral buoyancy. As the animal rises past its physiological depth boundary, the sudden drop in hydrostatic pressure and the rapid increase in water temperature cause its specialized ammonium-rich tissues to malfunction, leaving the animal semi-paralyzed and dying at the surface. When ancient sailors encountered these dying titans, the visual shock was profound. A giant squid floating at the surface can extend over 12 meters in length, with tentacles that undulate wildly in the waves like writhing serpents. The muscular mantle, stripped of its protective dark skin by scavengers, appears as a pale, ghostly white mass, easily mistaken for a submerged island reef. The central mouth of the creature features a massive razor-sharp chitinous beak, capable of snapping thick wood, surrounded by two enormous basketball-sized eyes that stared back at the sailors with an eerie, unblinking intelligence. When the dying animal performed its final desperate muscular contractions, firing water through its powerful siphon to escape back into the dark, it created a massive churning whirlpool that could easily swamp a small fishing vessel. Mariners returned to port with wild, exaggerated stories of these encounters, transforming a tragic, dying deep-sea invertebrate into an intentional, malicious demon of the ocean. A similar process of mythological inflation occurred across the ancient Mediterranean and Near East, giving rise to the biblical legend of the Leviathan. Described in ancient texts as a colossal, armoured sea serpent that breathed fire and ruled over the global oceans, the Leviathan represented the ultimate untamable force of nature. While the legend was heavily influenced by encounters with large coastal reptiles and whales, the specific anatomical descriptions of the Leviathan, particularly its elongated, ribbon-like body, its lack of traditional scales, and its sudden, dramatic appearances before major seismic events, point directly to another deep-sea giant, the Oarfish, Regalicus glesson. Living down to depths of 1,000 meters, the oarfish is a biological marvel that completely matches the structural outline of a classic sea serpent. It can grow to lengths exceeding 8 to 11 meters, making it the longest bony fish on the planet. Its body is bright silver, completely scaleless, and covered in a highly reflective coating of guanine that flashes brilliantly when exposed to light. Most striking of all is its dorsal fin, which consists of hundreds of bright blood-red rays that form a magnificent crown-like crest directly above its head, extending down the entire length of its undulating body. Under normal conditions, human beings never see a healthy oarfish. However, because the oarfish possesses a highly fragile, lightweight skeleton and lives near active underwater fault lines, it is uniquely susceptible to seismic disruptions. When an underwater earthquake occurs, the sudden release of intense low-frequency sound waves and toxic gases can stun or injure the fish, forcing it to rise to the surface. When an oarfish washes ashore or swims weakly near a coastal beach, its long ribbon-like body moves with a slow, serpentine undulation. To an ancient observer standing on the shore, seeing a silver red-crested serpent longer than a house emerge from the waves was an apocalyptic sight. The natural silver reflection of its skin was interpreted as glowing armour, and its bright red crest was transformed into a mane of living fire. By tracing these legends back to their biological roots, modern science does not diminish the wonder of these creatures. Rather, it enhances it. The true horror and beauty of deep-sea gigantism lie in the realization that the monsters our ancestors feared were not supernatural demons, but highly specialized evolutionary masterpieces. The Kraken is not a malicious destroyer, but a low-energy predator with basketball-sized eyes, designed to read the faint bioluminescent whispers of the midnight zone. The Leviathan is not an engine of cosmic chaos, but a fragile paper-thin silver ribbon that drifts vertically through the frozen dark with perfect thermodynamic efficiency. By decoding the science behind the myth, we bridge the gap between ancient maritime folklore and modern marine biology, proving that the deep ocean has always possessed the power to ignite the human imagination, hiding its greatest biological treasures behind the terrifying mask of the mythological unknown. To truly appreciate the horizontal expansion of deep-sea gigantism, we must examine the advanced technological systems that have allowed human beings to breach the abyss and study these magnificent titans in their natural habitat. For over 99% of human history, the deep ocean floor was a completely closed world, protected from human intrusion by miles of freezing water and crushing hydrostatic pressure. Early oceanographers had to rely on rudimentary iron chains, heavy hemp ropes, and blind scraping nets dropped from surface ships. These early sampling methods were incredibly destructive. By the time a deep-sea giant was dragged up past the extreme depth boundaries, the sudden, cataclysmic drop in ambient pressure and the rapid increase in temperature caused its specialized cellular membranes to liquefy, its proteins to denature, and its gasless tissues to expand violently. Scientists were left with damaged, semi-fluid remains that offered almost no insight into how these creatures actually lived, moved, and interacted in the dark. The true scientific revolution began in the mid-20th century with the development of manned deep submergence vehicles designed to withstand extreme physical forces. The pioneer of this technological shift was the bathyscaphe Trieste, a heavy specialized vessel engineered by Swiss physicist Auguste Piccard. In the year 1960, the Trieste achieved the ultimate milestone in deep-sea exploration, descending nearly 11,000 metres into the Challenger Deep within the Mariana Trench. The primary engineering breakthrough of the Trieste, and the subsequent generation of research submersibles like the famous Alvin, lay in the design of the crew capsule. To protect human observers from being crushed by over 1,000 atmospheres of pressure, engineers constructed a thick solid sphere made of forged titanium and high-strength steel alloys. Because a sphere distributes external physical force completely evenly across its entire surface area, it prevents the formation of localized structural stress points that would otherwise cause an implosion. To allow scientists to view the abyss, engineers replace standard glass windows with thick cones of optical-grade polymethyl methacrylate plastic, which actually presses tighter into the titanium framework as the hydrostatic pressure increases, creating an absolute watertight seal. Inside this artificial titanium bubble, atmospheric pressure remains completely constant at sea level, allowing human beings to sit safely within a few inches of a hostile world, watching giant amphipods and deep-sea sharks glide past through the dark. As technology matured into the late 20th and early 21st centuries, the high-cost, physical danger, and limited battery life of manned submersibles led to the development of robotic exploration platforms. remote-operated vehicles, and autonomous underwater vehicles. Remote-operated vehicles, such as the famous Jason and Doc Ricketts operated by modern oceanographic institutions, are heavy, robotic workhorses connected to a surface research vessel by a long, heavily reinforced umbilical cable containing fibre-optic wires and high-voltage power lines. This umbilical link provides the robot with an infinite supply of energy from the surface, allowing it to remain stationary on the abyssal plane for days or weeks at a time. These advanced robotic platforms are equipped with arrays of high-definition digital cameras that can capture images in low-light conditions using powerful arrays of light-emitting diode panels. This allows scientists to observe the true, undisturbed behaviours of deep-sea giants without blinding them with high-heat incandescent lamps. Furthermore, these vehicles feature highly articulate hydraulic manipulator arms lined with sensitive force feedback sensors. Instead of crushing a delicate deep-sea specimen, a robotic arm can gently scoop a giant isopod or a fragile glass sponge into a specialized insulated chamber known as a suction sampler or a biobox. These containment chambers are structurally insulated to maintain the cold, high-pressure environmental parameters of the deep sea during the long ascent back to the surface, ensuring that the specimens arrive in the shipboard laboratory with their delicate cellular and molecular structures completely intact. In the modern era of marine science, exploration technology has taken a radical microscopic turn, leaving behind heavy mechanical claws and moving into the realm of molecular genetics through the use of environmental DNA technology. Environmental DNA, or eDNA, relies on the scientific reality that every living organism continuously leaves traces of its genetic material behind in its surrounding environment. As a giant squid swims through the water column, as a Japanese spider crab molts its shell, or as a giant sea cucumber expels waste, it releases thousands of microscopic cellular fragments, skin cells, and metabolic fluids containing its unique deoxyribonucleic acid sequence into the water. Using automated high-volume water filtration systems attached to autonomous underwater vehicles, modern marine biologists can collect water samples from specific depth zones across the global oceans. Once these samples are brought to the surface, the water is forced through fine mesh filters to capture the suspended genetic fragments. Scientists then apply a laboratory technique known as polymerase chain reaction, which amplifies these minute degraded fragments of DNA, creating millions of exact copies until there is enough material to run through a high-throughput genetic sequencing machine. The resulting genetic data is then compared against massive international digital databases containing the known genetic signatures of thousands of marine species. This technology has completely transformed our ability to trace and catalogue deep-sea giants. Scientists no longer need to physically see or capture an elusive apex predator like the colossal squid to confirm its presence in a specific region of the Southern Ocean. By simply analysing a single litre of deep seawater, the presence of its unique eDNA sequence provides absolute scientific proof that the giant passed through that exact water coordinate within the last 48 hours. Ultimately, the union of pressure-resistant manned submersibles, high-powered robotic vehicles, and cutting-edge environmental genetics proves that human curiosity cannot be barred by the boundaries of the abyss. By engineering tools that mirror the structural resilience and efficiency of the very giants they are designed to track, scientists have turned the hostile, opaque void of the deep ocean into a transparent laboratory of discovery. These technologies ensure that as we continue to map the final frontiers of our planet, the hidden rulers of the deep will no longer remain nameless shadows in the dark, but will be recognized as the true biological masterpieces that they are. To fully comprehend the horizontal reality of deep-sea gigantism, we must examine the modern ecological stability of the abyss, a stability that is currently facing unprecedented anthropogenic disruptions. For millions of years, the deep ocean floor has been one of the most stable, unchanging habitats on Earth. The creatures that reside here, particularly the deep-sea giants, have evolved their massive body sizes, exceptionally long lifespans, and slow metabolic pathways under the assumption of absolute environmental permanence. Because their home experiences no seasonal shifts, no rapid temperature changes, and no sudden structural alterations, these animals have sacrificed evolutionary agility for long-term specialization. They possess almost no capacity to adapt to rapid changes. Today, this ancient isolation is breaking down as three interconnected human threats: deep-sea mining, global climate change, and ocean acidification, driven by escalating carbon dioxide emissions, converge upon the abyss, threatening to dismantle the delicate biological scaffolding that supports the largest invertebrates on the planet. We begin with the immediate mechanical threat of deep-sea mining. Across vast areas of the abyssal plains, particularly within the Clarion-Clipperton zone in the Pacific Ocean, the muddy seafloor is littered with millions of polymetallic nodules. These are small, potato-sized mineral formations that have taken millions of years to precipitate out of seawater, accumulating high concentrations of rare and valuable metals, including cobalt, nickel, copper, and manganese. These elements are highly sought after by surface industries to manufacture lithium-ion batteries and green energy technologies. The industrial process of extracting these nodules involves deploying massive, track-driven robotic mining vehicles that crawl over the soft sediment, scraping up the top layer of mud to harvest the rocks. This mechanical extraction causes direct, catastrophic destruction to the local benthic ecosystem. For deep-sea giants like the giant Isopod or the giant sea cucumber, the seafloor is not an empty desert. It is a delicate web of life dependent on the thin, undisturbed surface layer of marine snow. The heavy mining treads crush sessile organisms, destroy the ancient burrows of scavenging crustaceans, and permanently alter the physical density of the substrate. Furthermore, the industrial operations generate massive sediment plumes. As the mining machines process the mud and discharge the waste material back into the water column, they create enormous underwater clouds of fine silt that can drift horizontally for thousands of kilometers through the deep currents. These sediment plumes are highly destructive to the sensory and respiratory architecture of deep-sea giants. Filter-feeding organisms, such as giant glass sponges and massive tunicates, have their delicate mesh-like filtration systems clogged with sterile mud, leading to rapid suffocation and starvation. For visual predators like the giant squid, these dense plumes turn the crystal-clear darkness of the abyss into an opaque, muddy fog, preventing them from detecting the faint flashes of bioluminescent prey or seeing the approach of predatory sperm whales. The intense acoustic noise generated by high-power industrial machinery travelling through the dense water column further disrupts the vibration-sensitive statocyst networks of deep-sea cephalopods, leaving them disoriented and unable to navigate their home. Simultaneously, the overarching crisis of global climate change is beginning to penetrate the deepest layers of the water column. The primary engine of deep-sea circulation is thermohaline circulation, a global conveyor belt driven by differences in water temperature and salinity. Cold, oxygen-rich surface water sinks near the polar regions and slowly flows horizontally across the global abyssal plains, delivering fresh oxygen to the deep trenches. As rising atmospheric temperatures melt polar ice caps and warm the upper layers of the ocean, this vertical circulation pattern is slowing down. A reduction in thermohaline flow has direct devastating consequences for deep-sea metabolism. As the circulation slows, the replenishment of dissolved oxygen in the abyss declines, causing the expansion of hypoxia across the ocean floor. For deep-sea giants, which already operate at the absolute limit of oxygen availability, any further reduction in dissolved oxygen forces them into an evolutionary corner. While their high-affinity respiratory pigments like hemocyanin are masterfully designed to extract minute traces of oxygen, they cannot function if the baseline oxygen level drops to near zero. Without sufficient oxygen to power even their slow-motion basal metabolism, these giants will face localized extinctions, shrinking the geographical distribution of abyssal gigantism. The third and perhaps most insidious threat occurs at the molecular level, driven by the chemical absorption of atmospheric carbon dioxide into the global oceans. The world's oceans act as a massive carbon sink, absorbing approximately 25 to 30% of the excess carbon dioxide produced by human industrial activity. When carbon dioxide dissolves in seawater, it reacts with water molecules to form carbonic acid, which subsequently breaks down into hydrogen ions and bicarbonate ions. This chemical reaction drives down the pH of the ocean, a process known as ocean acidification. While ocean acidification is widely studied in shallow coral reefs, its impact on the deep sea is exceptionally severe due to a phenomenon known as the carbonate compensation depth. The carbonate compensation depth is the specific depth level in the ocean where the rate of calcium carbonate dissolution matches the rate of supply. Below this depth, the high hydrostatic pressure and low water temperature make calcium carbonate highly soluble, meaning it dissolves naturally in the water. Industrial ocean acidification is rapidly raising the boundary line of the carbonate compensation depth closer to the surface, causing the deep abyssal waters to become highly corrosive to calcium minerals. This chemical shift strikes directly at the structural integrity of deep-sea giants that rely on calcified exoskeletons for survival. The giant Isopod, for example, requires large amounts of calcium carbonate to reinforce its thick armoured chitin vault against the crushing physical force of the water column. As the surrounding water becomes increasingly acidic, the availability of free carbonate ions drops to near zero, making it biochemically impossible for the isopod to deposit new calcium into its shell after a molt. Its armour becomes thin, soft, and structurally fragile, rendering the animal vulnerable to internal tissue collapse under hydrostatic pressure and defenceless against predators. We see the same structural degradation in deep-sea clams, snails, and the calcified joints of the Japanese spider crab. Ultimately, the combination of industrial seabed mining, oxygen depletion from climate change, and the molecular stress of ocean acidification represents a profound environmental crisis. The magnificent giants of the deep ocean are not resilient to rapid change. Their strategy of living slow, growing large over centuries, and reproducing late in life makes them highly vulnerable to systemic collapse when their ancient, stable home is disrupted. If human activities continue to alter the chemical and physical parameters of the abyss, we run the risk of erasing these evolutionary masterpieces before we have even fully mapped their domain. Protecting the deep ocean is not merely a matter of conserving a remote wilderness. It is a vital imperative to safeguard the grand, ancient outlines of life's ultimate adaptations against the irreversible forces of human excess. As we ascend from the freezing depths and return to the sunlit surface world, we carry with us a profound new understanding of the planet's final frontier. The existence of deep-sea gigantism is not a biological error or an eccentric quirk of nature. Instead, it stands as the ultimate testament to the incredible resilience, flexibility, and adaptive genius of evolutionary design. In an environment engineered to destroy life through crushing hydrostatic pressure, absolute darkness, and chronic nutrient deprivation, nature has not merely found a way to survive, it has found a way to conquer. By altering their cellular membranes, slowing their metabolic processes to ultra-low speeds, and transforming their entire anatomical scale, these magnificent titans have turned a hostile void into a realm of long-term evolutionary security. The deep ocean is not a barren wasteland, but a vital, interconnected heart of the global biosphere that holds the answers to how life adapts to the most extreme physical boundaries in the universe. Protecting this timeless abyss and its hidden rulers is a definitive imperative for the future of our planet. If you enjoyed diving into the abyss with us, feel free to leave a like and subscribe for more deep-sea science. And before you go, drop a comment letting us know where in the world and what time it is as you are watching this video. Thank you for exploring the grand mysteries of the deep with us, and remember that no matter how dark or cold the world may seem, the spark of life will always find a way to grow, expand, and endure.